You are listening audio paper series with Yasir, and today's guest is Dr. Alikhan from Quadram Institute, Norwich, UK, to discuss one of his publication, SARS-CoV-2 variants of concern dominate in Lahore, Pakistan in April 2021. It is published in Microbial Genomics. Welcome, Nabil.
Hi, thanks for having me.
So, as the title states, SARS-CoV-2 variants of concern dominate in Lahore. What are these SARS-CoV-2 variants?
So this relates to SARS-CoV-2, which is the causative virus for COVID-19. And in particular, the variants of concern or the VOCs are defined by the WHO as particular variants of the virus that have either increased transmissibility, have been demonstrated to have detrimental changes in terms of the epidemiology. They are more virulent or have a more severe disease presentation, and in some way affect our ability to combat it in terms of public health measures or vaccines or drug treatments. So it's basically any variant that has some reason to be of concern, of something that we should monitor more closely than others.
Okay. So this relates to the mutations important for public health. How did you identify these variants of concern?
So these variants of concern have been defined through a whole group of different scientists working on it, and it's codified by the WHO. So they give them particular labels. The actual technical detection of them is done through amplicon-based sequencing of the virus, which we do using the Arctic protocol. And then based off those sequences, we can run the Arctic bioinformatics pipeline to first reconstruct the consensus sequence of each of the individual viruses in the samples.
Mm-hmm.
And then we can type those consensus sequences using the pangolin nomenclature scheme, which gives different lineage designations for all of the different sequences we recover.
Okay. So you have sequences of all the variants of concern. And when we have sequences from patient samples, then we can map them back to the variants of concern in our database.
Yeah, it's much deeper than just the set of variants of concern, which there are numerous numbers of lineages defined for SARS-CoV-2, and some of them do map across or are out of that huge pool. There are some selected as variants of concern. There is a lot of genetic variation in this virus. There is a lot of lineages from that, and not necessarily all of them are ones that those changes, those mutations do constitute a fundamental change in its behavior that we need to worry about each of them specifically.
Let's discuss this particular publication. What was the most dominant mutant you found in this study?
Yeah, so this study, the samples were selected from a short window, giving us a snapshot in April 2021. And at that time, we had 102 samples that we subjected to the process I just described before. And we found that the majority of them were the alpha variant of concern. So in the Pango lineage scheme, these are called B.1.1.7. And that likely has changes, that does change over time. In our paper, we recognized that there had been a change from the wild type to the alpha variant.
The last question. Do you think the sequencing and identifying different variants of concern could help in containing SARS-CoV-2?
Not in of itself. I mean, this is a very powerful tool that allows us to do a number of different things. It allows us to sequencing in of itself allows us to identify emerging variants that may possess mutations that allow vaccine escape or other such problems like that. They allow us to do traceback of outbreaks, so we can look at an outbreak, and because of the granularity of sequencing compared to other tools, we can differentiate when it is a spread across an outbreak that we can see the transmission across different individuals and may be able to say something about that. But ultimately, the utility of this tool has to be baked back into public health response. So this in of itself doesn't help anything. It needs to be used as a tool as part of a wider public health response to combat this pandemic.
Thank you, Nabil, for explaining the method details and important findings from this publication.